So very good evening everyone. I am Dr. Satyabrata, MDDM clinical pharmacologist, and today I am going to discuss on bioavailability and bioequivalence study. Coming to introduction, what is bioavailability? Bioavailability is the rate and extent to which the active ingredient of or active moiety is absorbed from a drug product and becomes available at the site of drug action. And what is bioequivalence? Bioequivalence is the absence of a significant difference in the rate and extent to which active ingredient or active moiety in pharmaceutical equivalents or pharmaceutical alternatives become available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed study. Essential to ensure uniformity in standards of quality, efficacy, and safety of pharmaceutical products. Both bioavailability BA and bioequivalence BE focus on release of drug substance from its dosage form and subsequent absorption in circulation. Similar approaches to measure bioavailability should be followed in demonstrating bioequivalence. Bioavailability refers to the fraction of administered dose that enters the systemic circulation in unchanged form. F is equal to denoted as F is equal to bioavailable dose divided by administered dose. Then factors affecting bioavailability of a drug. Physical properties of a drug, physical state, lipid or water solubility, dosage forms, that is particle size, physiological factors, ionization pH or fluid, pH of the fluid, GI gastrointestinal transit time, prolonged gastric emptying, increased peristaltic activity, fed state, fast pass metabolism, disease state, presence of other agents, enterohepatic cycling. This can also affect the bioavailability of a drug. Then coming to concept of equivalence. What is pharmaceutical equivalence? What is pharmaceutical alternatives? What is therapeutic equivalence? What is bioequivalence? You have to know all these things. So this is the picture showing how bioavailability is affected by patient related factors, administration related factors, strength of dosage form, excipients, other pharmaceutical factors, and also the bioavailability, the amount of drug released from the dosage form, amount of drug absorbed from the dosage form, then amount of drug in the body, concentration of drug in the central compartment, concentration of drug at the site of action, and then response. Then this is also showing patient related factor, administration related factors, strengths of dosage form, excipients, other pharmaceutical factors, in vitro quality control testing, amount of drug absorbed from the dosage form, then amount of drug in the body, in vivo bioequivalence studies, the concentration of drug at site of action, pharmacodynamic studies, clinical trials. Then coming to bioequivalence background, using bioequivalence as the basis for approving generic copies in US Drug Price Competition and Patent Term Restoration Act of 1984, Waxman-Hatch Act. CDSCO provides guidelines for BAB studies in NDCT rules 2019. Generic firms rely on findings of safety and efficacy of innovator drug after patent expiration. Innovator firms patent extension of 5 years to make up for time loss while they were going through FDA's approval process. Then this is the Orange Book, all FDA approved drugs listed, new drug application, abbreviated new drug application, and over the counter expiration of patent dates, Drug Price and Competition Act 1984, FDA required to publish approved drug products with therapeutic equivalence and evaluations. This Orange Book, approved drug products. Then establishment of BAB center. BAB studies should be conducted in BAB center having registration granted by Central License CLA Form CT08 for application, Form CT09 for grant. Inspection Rule 47 in Form CT09. Fourth schedule documents required for center registration according to GCP and GMP guidelines, good clinical practice and good manufacturing practice guidelines. The center should work. Then criteria and regulations in India for BAB study. Reference product type, reference product substitute, test product, then subjects, then strains, parent drug, fasting conditions, study pattern, IR, MR, drugs, fed state, immediate release, and MR, drugs, fed state, strength of dosage form, acceptance normal drugs, acceptance narrow therapeutic drugs, statistical data, demographic criteria, genetic phenotyping. Then requirement of BAB studies for IND investor new drug or NDA new drug application to establish equivalence between formulations used in clinical trials, stability studies, clinical trial formulations and to be marketed drug products, etc. ANDA abbreviated new drug application for generic drug products, change in components, composition and manufacturing process, changes in dosage form, capsules to tablets. Then objectives of BAB studies: development of suitable dosage form for a new drug entry, determination of influence of excipients, patient-related factors and possible interactions with other drugs, development of new drug formulations of existing drugs, control of quality of drug products, comparison of availability of a drug substance from different form or same dosage form produced by different manufacturers. Then bioavailability, parenteral solution, same active substance with same concentration, same excipient, then bioavailability may be done. Oral solution, same active substance with same concentration, excipient not affecting gastrointestinal transit or absorption, it can be bioavailable. Gas, in case of gas, if the substance is powder for reconstitution as solution, otic or ophthalmic topical solution, same active substance with same concentration, same excipient. Inhalational product oblique nasal spray, same active substance with same concentration, same excipient. In these cases, bioavailability can be done. Coming to methods used to assess equivalence: pharmacokinetic studies, pharmacodynamic studies, comparative clinical studies, dissolution studies. NDA new drug application versus ANDA abbreviated new drug application review process. NDA new drug application requirements: chemistry, manufacturing control, labeling, testing, animal studies, clinical studies, bioavailability. Abbreviated new drug application requirements: chemistry, manufacturing controls, labeling, testing and bioequivalence. Then coming to pharmacokinetic studies. 2-period crossover design. 2 formulations, even number of subjects randomly divided into 2 equal groups. First period, each member of one group receive a single dose of the test formulation, each member of the other group receive the standard formulation. Subjects 1 to 8, period 1, period 2, treatment standard. 9 to 16 subjects, period 1 standard, period 2 treatment. So this is a 2-period crossover design. Then Latin square crossover design for bioequivalence study of 3 drug products in 6 human volunteers. Subjects 1 to 3 4 6. Treatment for period 1 2 3 groups: A B A C B, 2 B C A C B A, 3 C A B B A C. Like this way the design is called Latin square. Then balance incomplete block design, BIBD. Subjects 1 2 3 4 up to 12 subjects. Treatment for period 1 2 group. 1 is A B C A B A C A D B C B D C D. 2 is B A C A D A C B D B D C. This is the balance incomplete block design, BIBD. Then another is parallel group design. Even number of subjects in 2 groups, each receive a different formulation. No washout necessary for drugs with long half-life. Treatment A, treatment B. Treatment A 1, 3, 5, 7, 9, 11. Treatment B 2, 4, 6, 8, 10, 12. Then this is the difference between parallel group and crossover study. In parallel study, groups assign different treatments, shorter duration, larger sample size, no carryover effect, does not require stable disease and similar baseline. But in crossover study, each patient receives both treatments, longer duration, smaller sample size, carryover effect, and requires stable disease and similar baseline. Then replicate crossover study design for highly variable drugs allows comparisons of within subject variances, reduce the number of subjects needed, 4 period 2 sequence 2 formulation design recommended, or 3 sequence 3 period single dose partially replicated. Then period group 1 group 2 1 TR 2 RT 3 TR 4 RT. This is the replicate crossover study design. Then pilot study. If the sponsor chooses in a small number of subjects to assess variability, optimize sample collection time interval, and provide other information. Parameters to be measured: Cmax, concentration maximum time, Tmax, time at which maximum concentration is shown, AUC, area under the curve. Then fasting and fed state condition. Fasting condition, single dose study, overnight fast 10 hours, subsequent fast 4 hours. Multiple dose study, 2 hours fasting before and after the dose. Then fed state studies, required when drug recommended with food, modified release product, assessment of Cmax, Tmax, difficulty in fasting state study, requires consumption of high fat food 15 minutes before dosing, provide 950 to 1000 kilocalories, fat 50%, protein 15 to 20%, carbohydrate 30 to 35%, ethnic cultural variation considered specified in protocol. Steady state only multiple dose study, long elimination half-life, toxic drugs requiring multiple dose therapy, some modified release drugs, combination products, drugs inducing own metabolism, drugs showing non-linear pharmacokinetics. Then parameters in multiple dosing studies, Cmax SS, AUC SS, C mean SS, fluctuation Cmax minus C mean. Then statistical evaluation, Cmax, AUC analyzed by using ANOVA, Tmax analyzed by non-parametric methods, use natural log transformation of Cmax and area under the curve AUC. Calculate 90% confidence interval for this GMR, geometric mean ratio, GMR for Cmax. Then calculate GMR for AUC, area under the curve. The calculated 90% confidence interval for Cmax and AUC, area under the curve, should fall within the range 80 to 125% range of bioequivalence. Then bioequivalence results 80% to 125%. Within 80% 125% demonstrated by equivalence. After 125% failed to demonstrate by equivalence. Before 80% failed to demonstrate by equivalence. Then bioequivalence assessment of two formulations of ibuprofen. There is published thing, and area under the curve, Cmax, Tmax, T-half, everything is calculated. Then pharmacodynamic studies, parameter studied, area under the curve, maximum response, time for maximum response. Then comparative clinical studies, comparative clinical study, artesunate suppositories and oral artesunate. Artesunate suppositories, oral artesunate, artesunate suppository 15 mg per kg per day for 3 days, oral artesunate 6 mg per kg per day for 3 days with mefloquine 25 mg per kg. 52 children participated. Large number. B studies 16. Then mean times to fever subsidence similar in two groups. Clinical parameters, cure rate by day 28 similar. time to parasite clearance not significantly more in suppository group, safety profile good in both groups. This is a comparative clinical study. Then coming to dissolution studies. Dissolution test USP apparatus 1 at 100 rpm revolution per minute or apparatus 2 at 50 rpm revolution per minute using 900 ml of the following dissolution media: 0.1 normal HCl hydrochloric or simulated gastric fluid USP without enzymes, a pH 4.5 buffer, a pH 6.8 buffer, or simulated intestinal fluid USP without enzymes for capsules and tablets with gelatin coating, and simulated gastric and intestinal fluids USP with enzymes can be used. Then dissolution studies: average dissolving time of pain relievers and dissolution times, types of pain relievers. Coming to conclusion, generics help patients by making drugs available at affordable price while retaining their quality. BAB bioavailability bioequivalence studies are growing day by day. Many BAB bioavailability bioequivalence CROs contact research organizations in India, translated into increased opportunities for pharmaceutical industries. So these are my references. NDCT rules 2009 is CDSU Central Drug Standard Control Organization. NDCT is New Drug Clinical Trial Rules. Then ICGCP International Council Harmonization Good Clinical Practice. And you can go through these documents to know more and more about bioavailability and bioequivalence and other things also. And for your further knowledge, and you can also go through the guidelines available in the online search. And please like and subscribe my channel, uh, and watch videos till the end of the topic and learn more and more about the disease and the drugs, and also other things that are given in my videos. And please like and subscribe, and you have to learn a lot of things from this channel. So that's all for today. Thank you very much. Jai Jagannath.